Welcome to the Science of Childhood Cancer virtual seminar series hosted by the St. Jude Children's Research Hospital Comprehensive Cancer Center. My name is Mike Dyer, and with Alberto Pelpo, I co-lead the Developmental Biology and Solid Tumor program here in the Cancer Center. So today, it is a real pleasure and an honor to introduce Ronnie George from the Dana-Farber Cancer Institute. Ronnie earned her MD degree from the University of Liberia and then her PhD from the University of Newcastle. She did her Pediatric Hematology Oncology Fellowship at Dana-Farber and worked with Tom Look. Ronnie is one of those amazing examples of a physician scientist who has transformed our understanding of neuroblastoma. She moves seamlessly between clinical, translational, and basic research. For example, in 2006, she published a first author paper in JCO describing a subgroup of neuroblastoma patients who had a more favorable prognosis. This, combined with other studies, has really led to our current risk stratification for this disease. In 2008, she had a landmark study in Nature describing activating mutations in ALK, and she went on in 2012 to publish her ALK mutant mouse model in Cancer Cell. And then more recently, she has dug into epigenetics and really started to look at enhancers and chromatin looping in neuroblastoma with a beautiful paper in Nature focused on the role of chromatin looping in resistance to ALK inhibitors. And so while I've always been impressed with Ronnie's ability to move effortlessly between the clinic, translational, and basic research arenas, the thing that I respect most is her continuous commitment to her patients. You can see through her papers and through her career, she always keeps the patients in mind and is always looking for ways to improve outcomes for children with neuroblastoma. So on behalf of Alberto Pelpo and myself and the St. Jude Comprehensive Cancer Center, thank you for sharing your research with us today, Ronnie, and thank you everyone for attending.
Thank you so much, Mike, for the kind introduction. I must say I'm really excited and honored to be here to present our recent work on neuroblastoma. So today I'm going to talk about something that we've been excited about, which is lineage plasticity in neuroblastoma, which is really the focus primarily of my lab. I have no relevant disclosures. And my talk today will be consistent of two parts. The first will be largely work that we published last year regarding the transcriptional and epigenetic reprogramming during therapy resistance in neuroblastoma. And the second part of my talk will be using cell lineage as a predictor of immune responsiveness in neuroblastoma. So just for those who don't think about this a lot, neuroblastoma, as we know, is a tumor of the developing neural crest, and it manifests as tumors in the adrenal gland and along the paraspinal region. It's really the most common extracranial solid tumor of childhood, with large number of cases in the younger age group. Although survival rates for lower stage neuroblastomas have improved, the survival of patients with high-risk neuroblastoma certainly lags behind. And neuroblastoma is a very heterogeneous tumor, clinically as well as genetically. Clinically, for example, this tumor has the ability to regress spontaneously in utero or during the first year of life. And at the other end of the spectrum, you have patients that come in with widely metastatic disease, with relentless progression and a poor survival rate. And in addition to clinical heterogeneity, this tumor is also marked by genetic heterogeneity. And one of the most enduring genetic aberrations is that of amplification of the MECAN oncogene. And this occurs in about 20% of neuroblastomas overall, and in about 50% of high-risk neuroblastomas. quite recently, it was understood, and we had certainly published, that MICAN is really governed by very large enhancer regions, also known as super enhancers, which can be seen in MIC amplified cells. And those, in fact, tend to amplify the expression of the genomically already amplified MICAN, thereby MICAN overexpression takes off over the transcriptional program of the cell. In addition to MICAN amplification, another mutational abnormality is that of ALK, where activating small molecule receptor sensitive mutations have been identified in neuroblastoma. And the combination of patients of the two abnormalities really leads to very aggressive tumors. So because of this, and because of the trials of ALK inhibitors in clinical trials, the current phase 3 Children's Oncology Group trial is actually testing ALK inhibitors in the clinic. And because we know that tyrosine kinase inhibitors can lead to resistance, we then decided to model ALK resistance in the lab and to study the mechanisms of such resistance. And this story was primarily through the work of very talented postdocs, David DeBruyne, and computational biologist Ruben Dries, both of whom have gone on to their own independent positions. So we used MICAN amplified ALK mutated neuroblastoma cells and develop resistance to the ALK inhibitor TAE684, which is the precursor of the Novartis ALK inhibitor ceritinib, with increasing concentrations of the inhibitor until they achieved quite stable resistance and had an IC50 of more than 30 times the sensitive cells. We ensured that these cells also maintained their resistance in vivo by injecting them into mice, and these tumor cells showed downregulation of ALK, showing that they were now no longer dependent on phosphorylated ALK, but had developed resistance to another mechanism. And again, from these resistant tumors, we generated cell lines and showed that they had still maintained their resistance, and that they were completely not responding to the ALK inhibitor. We also showed that these cell lines were also cross-resistant to two of the other inhibitors, the ceritinib, which is used in the clinic, as well as lorlatinib, which is also in clinical trials right now in phase 1-2 testing. So the development of resistance took a few months, actually, with increasing concentrations of the drug, during which time we observed that the cells underwent several morphological changes. They entered into a sphere-like, slow cycling state, during which they lasted for a while, and then after that they seemed to exit out of this state and become proliferative again. However, they look different from their original sensitive state. And we noticed that during this sphere-like state, they did express some of the early pluripotency markers, such as NANOX, OX2, and OX4, but did not really show increased expression of the sort of normal cancer or accepted cancer stem cell markers. So once we had developed the stable resistance, we went through all the different tests to look for resistance to tyrosine kinase inhibitors, For one thing, we had already established that phosphorylated ALK was no longer present, meaning that the cells were not dependent on ALK anymore. We did not see any secondary mutations, which is obvious from the lack of phosphorylated ALK. We did not see any activation of any of the other tyrosine kinases, which can also occur. And also, we didn't see upregulation of multi-drug transporter proteins either. With regard to MECAN, these cells also had amplified MECAN. In the sensitive and resistant cells, there really was no change in the MECAN status, and these cells retained the same amplified MECAN. And also, we did not see marked upregulation or change in any of the downstream signaling pathways that we observed. So when we looked at the gene expression signature of the parental and resistant cells, we observed that there was quite a transcriptional shutdown in these cells, shown here in this way, compared to the parental NMEC amplified cells, which were, transcriptionally very active. And because we had earlier shown that MECAN tends to cause upregulation of almost all the actively transcribed genes within the cell, so much so that if you if you knock down MECAN, you get a complete transcriptional shutdown. We then looked at MECAN expression, and we observed that MECAN was one of the genes that was downregulated in the resistant cells. We confirmed this by looking at MECAN RNA and protein expression, and then we identified that in the resistant cells, the downregulation of MECAN was really due to laying down of repressor histone marks, H3K27 trimethyl. In addition, genome-wide, when we looked at MECAN binding through chip-and-stick analysis of MECAN, we found that in the sensitive cells, as shown in black, there was MECAN binding to almost all over really the chromosome, as seen in this black binding, whereas in the resistant cells, the binding of MECAN had really gone down, except for its normal locus at chromosome 2p24. And that really corresponded to the expression of the number of active transcripts genome-wide, where there was a downregulation of transcription within the cell. We also noted that these cells were no longer dependent on MECAN expression. So then we looked at the minority of overexpressing genes in these cells, and we saw that one of the genes that took our interest was BORAS. There were also other lineage-dependent transcription factors such as 6-1, NeuroD1, NeuroG1, etc. And so we wanted to find out what was causing these cells now to grow and proliferate, albeit at a decreased rate as the sensitive cells. So we knocked down these genes in these cells and identified that really BORAS was the gene to which these cells seem to be dependent on. And again, we observed BORAS upregulation in our in vivo resistant models. We identified that BORAS was suppressed by promoter methylation in the sensitive cells, which was relieved in the resistant cells. And again, as I stated before, these cells seem to be dependent on BORAS because knockdown led to death. And the reason why we were interested in BORAS was BORAS seemed to be a paralog of the chromatin architecture protein CTCF. So we know that gene regulation in mammalian cells is achieved through CTCF partly. which together with the cohesin complex guides the organization of chromatin into large topologically associated domains or TADs, as well as functional loops that constrain contacts between a regulatory element and the target gene, or bring distant genomic segments into close proximity. And that there have been some really nice papers describing disruption of CTCF binding through somatic mutations or DNA hypermethylation, leading to loss of insulated boundaries and subsequent activation of oncogenes. So because of that, our interest in Boris was peaked. We noted that it was Boris and CTCF were very similar. They had this almost very homologous zinc finger DNA binding domains. Usually, its expression is restricted to germ cells, i.e., in the testis, as seen here, where its expression is usually cytoplasmic. It also functions as a RNA binding protein. So we looked at Boris expression in normal tissues, and as I said, is localized to the testis compared to CTCF, which is ubiquitously expressed. And then in the Cancer Genome Atlas, we noted that there was variable expression of Boris in certain cancers, but not in all cancers. And some of these in which we saw expression was non-small cell lung cancer and possibly Ewing sarcoma, where expression of Boris was related to high risk and metastasis and recurrence. In neuroblastoma itself, looking at various data sets, Boris expression was associated with high risk, high stage, and tumor progression. Associated, this is a picture of Boris staining in a low-risk tumor where, again, is primarily cytoplasmic, whereas in a high-risk tumor, it translocates into the nucleus. So because of its DNA binding function, we wanted to see whether it also bound to DNA in the resistant cells in which it was highly expressed. So we looked at Boris binding across the genome, comparing it with binding of CTCF, because we also wanted to know whether Boris replaced CTCF over the genome. And we found that in the resistant cells, yes, indeed, there was quite a lot of Boris binding compared to the sensitive cells. However, the binding of CTCF did not change between resistant and sensitive cells. Also, we looked to see where exactly Boris was bound, and we found that, again, Boris was primarily bound at enhancer regions through co-binding with H3K27 activation mark, and that there was increased Boris binding in resistant cells, as well as at the promoter regions, compared to sensitive cells. And along with Boris, there was also increased binding of BRD4 across the genome, as well as increase in H3K27 acetyl, as I said, but the repressor mark binding did not actually change in the resistant cells. And then we also noticed that when we compared the Boris binding with MICAN binding in these cells, in the resistant cells, there was much more of Boris binding compared to the sensitive cells, but also Boris primarily tended to occupy the MICAN binding sites that were present in the sensitive cells. And so there was actually, in a meta-gene analysis, the concurrence of Mikan loss and Boris gain at common genomic locations. And that was also translated into the expression of those genes to which Boris bound. In the sensitive cells, the Mikan bound genes were highly expressed, whereas in the resistant cells, this was supplanted by Boris. So because I had mentioned that Boris was a paralog of CTCF, we also wanted to know whether Boris took part in forming either structural or functional loops. And therefore, we performed a high-tip analysis of the parental and resistant cells. And we identified that Boris did promote certain loop interactions in these cells. The primary ones were promoter-promoter interactions, enhancer-promoter interactions, and CTCF promoter interactions. And we observed, again, that these loop formations primarily occurred between enhancers and promoter regions, and that there was not that much of concurrent binding of Boris with CTCF site. And this is just an example here of the Boris gene itself in sensitive and resistant cells. The blacks showing the sensitive, the red are resistant, and you can see that there is binding of Boris in the resistant compared to the sensitive cells. No real change in CTCF binding, but laying down of the enhancer histone mark HTK27 acetyl without much change in the repressor mark. And again, this is a picture of the high-tip analysis showing that there is loop formation even at the Boris site itself. So then we wanted to take a closer look at the types of genes that were bound by Boris and in which Boris formed a loop, a functional loop, and to see the expression level and the types of genes that were regulated in this manner. So first we parsed out the genes that were in the resistant cells that formed a regulatory loop with Boris. And from that end were led to increased expression. From that, we narrowed down the cells, the genes within the resistant compared to the sensitive cells, and were unique to the resistant cells, and were bound by Boris, but were also associated with large amounts of HTK27 acetyl enhancer histone marks, as well as BRD4, and we identified those genes that were highly expressed. Moreover, we then delineated those genes that responded to knockdown of Boris in these cells. And following this analysis, we came up with a list of 20 genes, some of which I'm showing you here, which all seem to be early neuronal lineage transcription factors. And these genes all had increased expression. They were all bound by Boris and formed a unique regulatory loop in the resistant cells, which were not seen in the sensitive cells, were also accompanied by a super enhancer and underwent a chromatin state switch either from neutral to open or close to open. And again, this is just showing you a picture of these particular genes that go from sort of open, closed chromatin to open chromatin. And furthermore, some of these genes really had Boris binding sites in their promoter regions within the resistant cells, but not really seen in the sensitive cells. So, and this is just an example of a track basically showing some of these genes in the sensitive and resistant cells. So for example, if one looks at the neuro-G locus in the sensitive cells, really they had no H3K27 or BRD4 or Boris binding. The main binding was with CTCF, whereas in the resistant cells, there was laying down of H3K27 acetyl, BRD4 binding, also Boris binding with a generation of multiple loop formation in the resistant cells. And the same goes for these genes 6.1 and 6.4. So next we wanted to know what the timeline of these events were during the development of resistance. So as I mentioned earlier, we had collected cells during the development of resistance, so that we had the sensitive cells, we had cells that were intermediate resistant and those that had fully achieved stable resistance. And so we then did single cell analysis of these genes, and so that they had really isolated into the three populations. The red being really the fully resistant, and then we had the intermediate resistant and the sensitive cells. And Disney analysis also subgrouped them into various different populations, and from that we were able to do a pseudo-time analysis which showed really the trajectory of the expression of genes during development. So the first thing that was inferred was decrease in expression of MIGAN, following which there was upregulation of genes such as SOX2 and SOX9, and the cells lasted or lived in that state for a while. And then halfway through the intermediate resistance was when Boris expression started coming up. And unlike the other genes like SOX2 and SOX9, whose expression fell with increase in Boris, ultimately there was increase in expression of some of these other neurogenes, but ultimately when stable resistance developed, Boris was the main gene that had the highest expression in the resistant cells that were subjected to single cell analysis. And again, as I mentioned before, the cells were dependent on Boris, and the knockdown of Boris appeared to also regulate these other genes and cause decreased expression of these genes. So our working model was that essentially there appeared to be chromatin remodeling and plasticity that occurred with development of resistance. So here we have the sensitive cells that are driven both by mutated ALK and MIGAN. And during the intermediate resistant cells, we felt that the cells had undergone plasticity to a very earlier state. And from that state, they had increased, slowly started increasing expression of Boris until Boris induced basically chromatin remodeling in these cells and drove the expression of a group of transcription factors through induction of functional regulatory loops in these cells. So essentially this story shows that these neuroblastoma cells can undergo plasticity with stress, and that this especially occurred in terms of this particular resistance model, and that Boris, like CTCF, is paralog, is capable of inducing new loop interactions in cells. And so with this, I'm just going to stop for a couple of minutes for questions.
Okay, thank you, Ronnie. I have a quick question, and then there's some coming in as well. I guess my first one is, if you look at the high-risk neuroblastomas from the patient samples that have high levels of Boris in the nucleus, what percentage of those have a decrease in MYC-N expression?
Yeah, that's a really good question, and we haven't actually looked at that. We're continuing to study resistance models to see, at what point does MYC-N go down, and also looking at these the expression data sets to see to look at the MYC-N expression levels in those. So that's something we're still looking at.
And then a question that's come in is, how are you defining resistance? So are you thinking of clinical sort of regression resistance, histologic, or something else? And I know the cell culture experiment is very different from sort of in patients, but how are you going to tackle relating those two processes?
Yeah, so I think that the analogy really is in patients who respond and respond to initial treatment. As we know, in neuroblastoma, one sees a relatively early response, but then they always patients always tend to relapse. And the theory is that a percentage of cells undergo tumor plasticity and sort of revert back into a fully drug-resistant state. And then once the initial stress is over, and that can last like months or even a year or so, these cells then slowly start coming back up. And the important point is the lineage of these cells are completely different from what was seen originally in the initial diagnosis.
Fantastic. Why don't we continue on, and then as other questions come in, we'll tackle those at the end.
Okay, thank you. So I want to talk about the story that we're really excited about. It's completely unpublished, and these were done by two phenomenal postdocs in my lab, Satyaki, who did all the cellular work, and Sanjukta, computational biology postdoc, who did all the analysis. So we know that in neuroblastoma, as is the case with a lot of the pediatric solid tumors, the successes of immune therapy like checkpoint inhibitors haven't really been translated into the solid tumors. People have tried various clinical trials, etc., but these have not been very successful. And neuroblastomas are generally thought to be immunologically inert or cold. However, we know that the anti-GD2 antibody therapy, which relies on immune-mediated cytotoxicity, is effective in a lot of the high-risk patients and is actually standard of care. So this suggests that certain neuroblastomas at least could benefit from other forms of immunotherapy. However, treatment with cytotoxic CD8 lymphocytes or CAR T cells or checkpoint inhibition have really produced suboptimal results. And so there are major impediments to the success of immune therapy. And this is a slide from review on neuroblastoma, and this essentially shows the different ways in which the immune response to neuroblastoma can be enhanced. However, both the neuroblastomas can generate what's called tumor intrinsic mechanisms to evade immune response, as well as immune suppressive mechanisms within the tumor microenvironment. So for example, in neuroblastomas, you could have a decrease in MHC class 1 molecules, defects in antigen processing machinery that render these cells resistant to T-cell cytotoxicity, downregulation of NK cell ligands, upregulation of CD47 to avoid macrophage-mediated phagocytosis. And then again, within the microenvironment, we can have T-regulatory cells, myeloid-derived suppressive cells. Both of these actually lead to decreased T and NK cell infiltration, homing of immune cells, and difficulties with recognition and result in cytotoxicity. One of the major aberrations in neuroblastoma, which I talked about earlier, is MECAN amplification. And MECAN amplification generally is known to lead to evading immune destruction. For example, MECAN amplified tumors specifically downregulate MHC molecules, and these are associated with poor infiltration of cytotoxic T cells and reduced expression of NK cell ligand. However, MECAN amplification is seen in 20% of neuroblastomas, and it's not really known, what really dictates the 80%, the remaining 80%, and whether they even have the capacity to induce a productive immune response. So we wanted to look at this question, and so we took a published data set of expression from primary neuroblastoma tumors of about 498 tumors that were really well represented in terms of the MECAN amplification status. There were about 98 tumors, age, INSS stage, as well as risk. This data set was very well annotated, and we performed analysis to quantify the tumor-to-tumor gene expression variability and cluster tumor types based on gene expression profiles. So basically, in this unbiased analysis, all tumors within one cluster would share similar gene expression profiles while being dissimilar to those within other clusters. And so we first identified the 500 highly variable genes on the premise that these would be the most likely to contribute to distinct molecular subtypes. And then the data were dimensionally reduced by principal component analysis, and the top 20 PCs were selected for clustering. And clustering was done and visualized by nonlinear dimension reduction tool, similar to TISNI, but this time we use UMAP. And to explore the transcriptional differences, we looked at the differentially expressed genes within the clusters that arose from this clustering. And so what we identified was 4 different clusters that out of these 498 tumors. So looking at the genes that were expressed in these clusters, so for cluster 1, for example, appeared to have so-called genes that were involved in cell proliferation, biosynthesis, and appeared to be sort of what one would call MECAN target genes. And this cluster segregated primarily with MECAN amplified tumors, and therefore we named it cluster 1. And you can see this cluster 1, when you overlaid it with the other clinically and associated prognostic factors, cluster 1 appeared to have a preponderance of NMIC amplified tumors. They were all over the age of 18 months, primarily had stage 4 and were classified as high risk. So the second, and these are just some of the genes that were represented in this cluster. For example, MECAN is target, ornithine decarboxylase 1, etc. The next cluster, which was enriched for genes in neuronal function and nervous system development, we termed neuronal. And cluster 3, which accounted for about 28% of the tumors, were enriched for genes that were involved in immune function. So for example, interferon gamma mediated signaling pathway, regulation of T cell, and these we termed immunogenic. And cluster 4 seemed to be far apart from the rest of the clusters, and they comprised primarily of stage 4S tumors and were enriched in genes involved in fatty acid and cholesterol homeostasis and were termed metabolic. So the neuronal and the metabolic clusters, the neuronal and metabolic clusters, tended to occur primarily in children who were less than 18 months of age. That's these two. And those who had also stage 1, 2, and 3 tumors, while the remainder, the immunogenic cluster, the one that was of interest to us, appeared in children over the age of 18 months, were primarily non-MECAN amplified and had a combination of high and low risk tumors, but also had predominantly metastatic disease. So our analysis came up with 4 of these clusters. So then to pursue the genes that were present in the immunogenic cluster, we looked at a number of genes. And for example, in the top layer, we saw that there are certain genes that were associated with immune activation were really represented in the immunogenic cluster, but at the same time, genes that were involved in immune evasion, like LAG3, TIGIT, FOXP3, were also represented in this cluster. So that made us look through the remainder genome-wide, and in order to actually quantify the immunogenicity of these tumors. So we generated an immune activation score based on the relative expression of a curated set of genes known to have major roles in tumor cell intrinsic immune functions, like antigen processing machinery, regulation of MHC expression, NK cell ligand expression, etc. And once we assigned a score to each of these MECAN non-amplified tumors and arranging them in ascending order, we observed that a significant number with the highest immune activation scores really fell predominantly within these immunogenic clusters shown in green. And also, because a cytotoxic immune response is usually accompanied by immune suppression or evasion, we also developed an immune evasion score, again, based on the relative expression of immune evasion genes, most of which were markers of T cell dysfunction. And again, we see that they were represented primarily in the immunogenic cluster. So essentially, there is a subset of tumors within the non-MECAN amplified neuroblastomas that have both immune activation and immune evasion gene expression from the cells, pointing to their ability to induce an anti-tumor response. So then we wanted to know, was there any kind of biomarker that we could use to identify these tumors that possibly could respond to immune therapy? And we looked at the known prognostic markers that are known in neuroblastoma, for example, age, stage, MECAN amplification, but none of these appear to be significant. And so and then it's known that certain cell states associated with stemness and senescence can affect T and NK cell mediated anti-tumor immunity. And so we then asked whether neuroblastoma cell state could be associated with tumor cell immunity. And about 3 years ago, two independent groups from Amsterdam and France, Rogier Vertig and Olivier Delache and Isabelle Jenny-Leroussi had identified two distinct cell states in neuroblastomas, a differentiated sympathetic neuron-like state, also known as adrenergic, defined by these lineage markers, including FOX2B, TBH, and TH, and a mesenchymal or neural crest-like state characterized by expression of mesenchymal type genes. And so we wondered whether the immune response would be related to cell lineage. And so to test this prediction, we first quantified an adrenergic and mesenchymal scores based on the lineage signature established by Groningen and al. In the group of Rogier Vertig in one of the papers that were published. And we assigned an adrenergic or mesenchymal score to each tumors within our previously identified 4 clusters. And we saw that there was significant enrichment of the mesenchymal cell state within the immunologic clusters and the metabolic clusters. By contrast, the high MECAN state and the neuronal state were really enriched for adrenergic lineage type tumors. And so in the interest of time, I'm not going to go through this whole analysis, but using the relative mesenchymal score for each tumor and further analysis, we identified that really the significant predictor of immunogenicity was the amount to which, or the degree to which, the tumor appeared to be mesenchymal as opposed to adrenergic, because tumors with high mesenchymal scores or high appeared to be represented in the immunogenic clusters compared to tumors with low mesenchymal scores. We then wanted to know whether the tumor cell intrinsic gene expression also, that we noted in the mesenchymal tumors also associated with tumor cell infiltration into the tumor microenvironment. And so we used 2 orthogonal approaches. So first we looked at established signatures of immune cell infiltration based according to the mesenchymal minus the adrenergic score. And we observed that tumors that had high mesenchymal scores had also overexpression of immune cell infiltration into the tumor microenvironment compared to tumors that had lower mesenchymal scores. And in addition, because through this, we understood that M-A score was a better predictor of immunogenicity. And in our multivariate multiple regression model, we also looked at M-A score, mixed score, as well as mixed signature to assess the independent significance of this. In addition, we also use CyberSort as a deconvolution approach to estimate the immune cell fraction associated with these adrenergic and mesenchymal tumors. And we saw that in the most mesenchymal of tumors, there was increased infiltration of cytotoxic CD8 cells, as well as an increase in regulatory T cells, also with cells expressing markers of T cell exhaustion. So ultimately, this showed that through our analysis of primary tumors, tumors that were more mesenchymal had evidence of T cell inflamed infiltrate. So next, we wanted to see whether this occurred and was primarily intrinsic to the tumor cells or conferred by the tumor microenvironment. And when we looked at the lineage identities of a number of cell lines and classified them based on immune activation score, again, we saw that the more adrenergic cells tended to have lower score compared to cells of the mesenchymal lineage. And that was shown here in terms of expression of antigen processing machinery genes in these cells. So in order to do further analysis, we looked at a particular cell line, SKNSH, was established several years ago, but has developed into two clones, and they were divided into two clones based on their morphological phenotype. There was the SHSY5Y cells that were neuroblastic in nature, and Chep cells that had substrate adherent morphology. And the prior two groups had already identified that the 5Y cells were adrenergic, while the Chep cells were mesenchymal. And using our RNA-seq analysis of expressed genes, we identified that the mesenchymal cells express most of the HLA antigens as well as ligands for NK cell receptors. We then noted that PRRX1, a lineage-specific homeoblox transcription factor that is highly expressed in the mesenchymal cells, could be used to program adrenergic cells into the mesenchymal phenotype. And overexpression of PRRX1 into the adrenergic cells led not only to expression of mesenchymal genes, as was shown by these two groups, but we also noted overexpression of tumor cell intrinsic immune genes, and especially increased expression of HLA and NK cell ligands, which were presented on the cell surface. In addition, we also showed that in resistance models that we had developed in the lab, that had acquired the mesenchymal phenotype during resistance, these cells again tended to express immune marker genes, again the HLA antigen genes, as well as ligands for NK cells. Next, we found that in the transition from the adrenergic to mesenchymal phenotype, these genes that had re-expression, especially the immune genes, had developed re-expression, were epigenetically regulated. So what we saw was that among the antigen processing genes, there was increased deposition of the activating histone marks, whereas decreased deposition of repressor histones. And that we saw in a number of different genes, not only in our resistance models, but also in the cells in which we had induced the mesenchymal phenotype by overexpressing the PRRX1 genes. And we also saw it in spontaneously evolving resistance. And again, we saw, because of the increased deposition of the repressor mark in the adrenergic cells, we looked at the PRC2 complex and found that these genes were heavily enriched with members of the PRC2 complex, including EZH2 in these genes. So I don't know how much more time I have, Mike, so.
Yeah, I think we should probably formally wrap up, and then, as I said, we can take some questions and discussion afterwards. So if you could maybe summarize, that would be fantastic.
Yeah, so I think the last part of it is that we showed in vivo that the mesenchymal cells were capable of engaging cytotoxic T cells, as well as the mesenchymal neuroblastoma cells were able to be activated by, were activating NK cells and induce degranulation and endocytosis in NK cells, and that this effect was increased by concomitant treatment with EZH2 inhibitors. So the summary of this part of the talk is that through an unbiased analysis, we identified that a subset of MICA non-amplified neuroblastoma is characterized by the presence of activating and repressing immune gene signatures, meaning that they're capable of eliciting an immune response. And these tumors consist predominantly of cells of the mesenchymal lineage, and that the expression of these is really epigenetically regulated, and that PRC2 inhibition can induce this immune gene expression in cells of the adrenergic lineage. So finally, I just want to thank all our collaborators, especially Judy Lieberman's lab at Children's for help with the immune studies, members of my lab, especially those that really contributed and worked on these projects, as well as our funding societies and foundations that have been especially generous during these times, and lastly to our patients and parents. Thank you.
Thank you so much for an amazing presentation. And thank you for taking a break in the middle. I know that cut away from some of your time, but I think it's great to answer questions along the way. So we have several questions. I've been putting them together. We'll go ahead and get started, and then I'll check back on the chat on a few additional. But thank you, everyone, for coming to the St. Jude seminar series on childhood cancer, and we look forward to seeing you again in the future. If you'd like to stay on for additional discussion, we'll wait a minute or two for folks to log off, and then those that want to remain on will continue to address questions through the chat. That was great, Ronnie. Thank you so much. Has that immune profiling study been published yet, or are you guys in the final stages? Looks like a pretty complete story.
Yeah, so we just submitted it. So
exciting.
Yeah.
And you know, while we're waiting, just again for folks to log off quickly, what role do you think cellular heterogeneity plays, maybe even in both studies, right? Both the Boris study and the immune study, right? If the tumors are really a mixture of cell populations, I know it's hard to tell from your bulk analysis, but how are you thinking about that?
Yeah, so I think that again, we need to do many more studies of single cell sequencing,
Hmm.
primarily of primary tumors, but also, and I know this is hard, but to acquire specimens, but really for relapse cases, because
Yeah.
I think that's where the tumor heterogeneity is showing up the most. And really, I think that um, the cells these are tumors that have undergone chemotherapy, radiation, etc., and they have completely metamorphosed into relying on other um, oncogenes or other regulators to continue their proliferation. So I think that sort of relying on initial diagnosis to treat patients is probably suboptimal.
And do you have any examples where at diagnosis and relapse, there's a complete shift in the sort of transcriptional phenotype?
Um, we
It's
we
hard to,
don't because again the issue is the paucity of samples to
yeah,
do studies. As we know, in relapse cases, usually most people don't do biopsies, especially since relapse usually occurs as skeletal relapse, and it's doubly hard to obtain relapse samples. So
yeah, yeah.
but if one compares other tumors or adult cancers, essentially that seems to be the trajectory of the tumor or the natural progression of the tumor.
Terrific. Okay, so we have several questions. I'm going to break them into part one, part two, and then there were some more general kind of therapeutic questions, which will play to your expertise with patients. So in part one, they were asking, how exactly is MYC downregulated? Is it through the enhancer? And then does it matter if it's MYC amplified through tandem repeats on the chromosome or through double minutes? Or you've not had a chance to kind of dig into that yet?
Yeah, we haven't had a chance to look into that second part, but MYC is downregulated through laying down of repressor histone. So the whole locus was enriched with H3K27 trimethyl in the resistant cells.
Okay, so enhancer and promoter specifically.
Mm-hmm.
Okay, great. Did you have a chance in your analysis of Boris high and Boris low tumors? Were there any of the older ATRX mutant AYA patients in that cohort?
Um,
Just
we
curious if
didn't
that looks different.
specifically
Yeah,
look, yeah, but we can check.
I want to go check now. Yeah, it'll be interesting to check. They're hard to come by, and the COG cohort didn't have a lot, but we can look through some of the St. Jude Cloud data and see what Boris expression looks like there.
Yeah.
In part two, let's see. Okay, so you already addressed the question about cellular heterogeneity, thinking about part one, but also part two in terms of the immunotherapy. Do you think this immune signature, I don't know all the genes and pathways, do you think that's relevant for both NK cell mediated therapy, which you pointed out is standard of care now, or anti-GD2 immunotherapy, which is thought to be mediated by NK cells, and potentially down the road for maybe CAR T cell, therapy for maintenance or something like that for neuroblastoma? Do you think that immune component is relevant for both?
I think what we saw in our studies were primarily the effect on NK cells. That tended to be very, very robust. So we think that the reason for the T cell results not being as good as those of the NK could be, other factors that promote immune evasion. But with the the NK cells, it was very clear, especially for the NKG2D receptor on NK cells. We found that their specific ligands, which are MIC-A, MIC-B, and ULBPs, they were suppressed on the adrenergic neuroblastoma cells. They had suppressed by PRC2 deposition. And if you were to relieve that either by EZH2 inhibitor or by converting them to the mesenchymal phenotype, they started expressing these NK cell ligands, and we show that they go through all the different stages of NK cell recognition, degranulation, and receptor endocytosis.
Okay, great. Question just came in. How does the ex vivo study correlate or relate to congenital neuroblastomas that regress naturally? Or I guess it's the 4S's, what is different there? Is that just completely different biology?
Yeah, so I think, there are several things that are clear from the second part of the talk. We found that among the tumors that were most immunogenic, apart from those in the immunogenic cluster itself, were the ones in cluster 4, which involve primarily the stage 4S tumors. So it kind of lends credence to the theories about, the spontaneous regression being antibody mediated in a way. And we were also gratified to see that the GO categories showed, those involved lipid metabolism. And I know that there was a publication beforehand where 4S tumors were found to be especially enriched in those in cholesterol biosynthesis and lipid metabolism. So, yeah, so that's further work needs to be done, of course, but
always.
It would seem as though the spontaneous regression would most likely be antibody immune mediated.
Terrific. And then there was just a couple of folks asking for asking questions about the clinical management, and just trying to understand how this disease is managed under certain circumstances. So I'll throw those both out to you. I think it'll be useful for the audience. So one question is, what role does surgery play for high-risk tumors? So for example, if you have a patient with high-risk neuroblastoma and you believe you've gotten all the primary lesion with surgery, what is the prognosis, and is it likely they have distant mets that you just can't detect and then they go on to have a poor prognosis, or can you cure those patients with surgery and maybe local radiation?
So are we saying this surgery alone? Because
well, with chemo, but if you get the primary lesion, even for a high-risk patient, what's the overall outcome?
So I think, there were some studies earlier on that showed that the extent of surgical removal really correlated with outcome. But I think we know that, after induction therapy, very good partial response, obviously portends a much better prognosis than if you have remnants of tumor. And again, the complete resectability of the tumor would actually go towards improving survival, but ultimately through several different iterative trials, we find that alone really isn't sufficient. Chemotherapy, local surgery, stem cell rescue, local radiation, but also, the dinutuximab plus cis-RA also plays a major part, and that really is the standard of care for COG. And even with that, survival rates have improved, to about 50% or so. But again, relapse rate, especially in high-risk tumors, relapses are invariably fatal. So, and while surgery and the extent of the removal of the tumor helps, that alone is not sufficient. And usually when kids relapse, they almost never relapse at the primary site. So it's usually in
Right.
distant metastatic site. So
Yeah, unfortunately. One final question. DNA methylation arrays have been extensively used in brain tumors for classifying and stratifying patients, and are starting to be applied to solid tumor patients. Do you think, or maybe you've done some preliminary studies, that DNA methylation may eventually be used to identify tumors with the adrenergic mesenchymal immune signatures, right, that you sort of looked at the gene expression profiles, and that going forward, that might be a way to start thinking about stratifying patients to treatment arms?
yeah, I think that, that really is a very good point. And it is possible that that might be what is used. I think ultimately with all the wealth of information, the genetic information that's coming up,
Right.
we need to sort of arrive at some sort of easy test so that we are able to stratify patients. It could be methylation arrays. It could be a signature, or it could even be, a sort of simple IHC, so expression of a protein like PRRX1 or preponderance of multiple antibodies that people, not just in the States, but in other countries can use easily and cheaply, and without the use of a lot of tumor tissue. So.
That's right. That's right. All right. Well, thank you so much, Ronnie. We're grateful. Fantastic stories. If you don't mind staying on the line just for a minute, I'd like to chat with you offline. And then for everybody else who stayed on for the discussion, thank you so much for joining us, and we'll see you at a future seminar. Thank you.
Thank you.